Hello, everyone. So thanks for joining this webinar that is organized by the Phytobiomes Alliance. So I am Claire Rogel-Gaillard from INRAE in France, and I chair the Animal Microbiomes Working Group, and it's my pleasure to introduce this webinar series. So as you may know, Phytobiomes Alliance is a non-profit consortium of industry, academic and governmental scientists. So it is an international alliance for phytobiomes research. And we thank the different sponsors. And as you may see, I'm a member of INRAE, and INRAE is also a sponsor. So what is the vision of the Phytobiomes Alliance? So the vision is that by 2050, all farmers have the ability to use predictive and prescriptive analytics based on geographical and biological conditions for determining the best combinations of plants management practices and inputs for a specific site in a given year. And so it is a matter of plants, but also management practices. And as you see on this picture, there are many different topics that are included in the Phytobiomes Alliance vision. So here you have this combination of plants, soil, the climate change, the weather, the water, and so meaning that there are connections with the resources, and it is connected with all the living organisms. And the phytobiomes comprise all the microbiomes and microorganisms, microfauna, and the connections with the living organisms like arthropods, other animals and plants. And of course, it is also modulated and influenced by management practices. Few examples of phytobiomes topics: crop fields, pasture, vegetable garden, forest, vertical farm. So that's only a subset of examples. So here today we are in the frame of the Animal Microbiomes Working Group. So there are other working groups, and you have the 4 other working groups on this slide. So we try to lead and coordinate efforts on specific topics, and animal microbiomes are very connected to environmental and also plant microbiomes. So the aim of this working group is to identify knowledge and resource gaps to advance our understanding of the interconnections between people, animals, plants, and their shared environments as defined in the One Health approach. And you have this scheme combining plants, animals, humans, environment in One Health vision. So just to let you know that so it is a webinar that we are proposing today, and the next one will be on June 6th, 2025, and the theme will be 100 years of collecting biological materials, the diversity of the ATCC collection, and the presenter will be Victoria Knight-Connolly from USA, and you can already register. So this webinar will not be organized by the Animal Microbiomes Working Group, but by the Phytobiome Alliances as a whole. So feel free to register from now. So if I go back to what we are doing today, so the webinar is recorded and will be posted on the Phytobiomes Alliance YouTube channels later today. So you will have the link to have access to this record. The presentation section will be followed by a question and answer session. So for the questions, you have to submit your questions in the Q&A panel, and you can also use the chat. And you have also, in the end, you will have a PDF of the slides that will be presented from the chat panel, and you will have also access to few references that are cited during the presentation. And if you want to register to the mailing list and receive news about events organized by the Phytobiomes Alliance, feel free to do that. And here you have this link to register. So today it's my pleasure to introduce 2 colleagues from INRAE, Jordi Estelle and Catherine Larzul. They work at INRAE in France. And the topic and the title of their talk is Beyond the Host Genome, host genetics and gut microbiome determinism in livestock. And Jordi and Catherine will share the floor and present a very fruitful collaboration on the holobionts in animals. So I'm stopping to share my slides, and Jordi and Catherine, feel free to share your screen and start your presentation. Thank you very much. See you for the Q&A session, and you have 40 minutes to present your talk.
Okay. Thank you, Claire. So I'm going to share the screen here. Do you see it okay? You see also the mouse? Perfect. Okay. So thank you very much for the opportunity to present our results here in the Phytobiomes Alliance. So as Claire was said, so we are Catherine Larzul and Jordi for presenting the results on beyond the host genome, host genetics, and gut microbiome determinism in livestock. Okay, good. The first slide is just to detail what are these holobionts. So there are two images on the screen. One, it's a bit more explicit in the right with the concept of the host genome plus the microbiome, which makes the hologenome. This is the genome combined of the host and the microbiomes, and these organisms here represent the holobiont. And then you have also the environmental metagenome, and it's all this in here that is it's the analysis of the Phytobiomes Alliance. And then in the left you have a beautiful image that it's trying to see the same thing with humans and a human in the left and a human in the right that's looking at each other and wondering which is here the host, the genome, or not. This is the holobiont. So the diversity and the determinism on the whole of the animal microbiomes it's very large, and it's not the scope of this presentation here, but just to show you that there are very different microbiomes in an organism, like here in a woman with many different compositions of microbiomes in function of the organ, and there are also many different determinants of the diversity and the composition of these microbiomes. And here you have in an image for the pig in a beautiful review that it's the microbiome of the pig and the intestine microbiome is going to be impacted by the diet, the health, the age, the housing environments, the use of antibiotics and probiotics, and also, of course, the host genetics. And this is the topic of the talk today. How the host genetics is going to impact digestive microbiomes in livestock. So why are we interested on this digestive microbiota? So it's essentially because it's associated with phenotypes of interest for livestock production, like can be feed efficiency, health, robustness, growth, welfare, or even the environmental impact. So there is a significant impact of host genetics on composition and diversity of microbiota. And we know this already thanks to estimations of heritability, genome-wide association studies, and there are even causal mutations identified for a few cases. So this is true for a lot of livestock that we are studying in the community, and the question is how we can use this later on. And for the presentation today, we're going to present results that we generated in our institute in pigs and also in cows. So for the pigs, we start from previous results published a few years ago already, that we showed there that the pig fecal microbiota ecosystem is organized in two major enterotypes, two types of ecosystems. One enterotype that is characterized with abundance of Prevotella and Mixucella, and we see here in the clustering analysis in blue. And then another enterotype in red that's characterized by an abundance of Ruminococcus and Treponema. So this was done with more than 500 animals at 50 days of age. And the question here is well, what is an enterotype? It's a type of the ecosystem, which is an ecotype, that is a group of organisms that is adapted to a specific environment. And here's an example from a forest. You can see on the left a forest of one type, we could say ecotype 1 or ecotype A, and another forest in the right. They are both forests. They share many characteristics, but they are slightly different species and slightly different also properties in here. So for the enterotype, we apply this concept to the microbiota ecosystem in the gut. And the question that we are posing here in our research is that given that the host genome impacts microbiome composition, we try to answer it if it is possible to use this host genetic selection to influence fecal microbiota composition. And from the previous results on the enterotypes in pigs, so the first question was if we could find K-driving taxa from the network interactions, and here we confirm in here that every node represents a taxa, a genus. We can see here that there is a strong correlation in green between Mixucella and Prevotella, which are both correlated negative with Ruminococcus and Treponema. And they are also well connected in positive on each of the sides of the graph. So it means that these four taxa can be useful key-driving taxa for applying the genetic selection in here. So I give the floor to my colleague Catherine Larzul, who will explain you how we set up the genetic selection experiment to implement this selection.
Thank you, Jordi. So here, because the idea was also to prove that it's possible to modify microbiota composition by selecting the host the old-fashioned way. That means you look at the host, you look at the performance of the host, and then you try to select and change the phenotypes. So in this experiment, we really considered the enterotype as a phenotype of the host, but for genetician, it's nearly impossible to select on a kind of enterotype, either because if you look at A and B, it's very difficult to select among a lot of A and a lot of B. So we had to find a different way for selection. And here what is represented here is really we limited the information used to the 4 genera identified as key taxa for the 2 enterotypes. So it will be Prevotella and Mitsuokella for one possible selection, and Treponema and Ruminococcus for another kind of selection. These genera were identified in previous studies as quite heritable in the genetician quantitative genetics theory, and the frequency, the occurrence, the relative abundance of each genera was sufficient to provide variability for selection. So we begin a selection experiment. So we constituted what we call a foundation pool of animals. So it's written on the slides. It's more than 300 piglets from a largely diversified population, the most possible for us to provide. So they were from 30 liters. And there were, we collected the feces, and from the feces, we extracted the bacterial DNA, and we sequenced 16S RNA 16S. And it was possible with relative abundances of the genera to begin this selection. So we selected boars and sows with higher relative abundances of Prevotella and Mitsuokella to constitute what we call the HPM line, and we selected boars and sows with higher relative abundances of Treponema and Ruminococcus, but mainly Treponema to constitute the line HRT. And just you see that from this base population, the frequencies of enterotypes. So you have to make a difference between the lines and the enterotypes. So in the base population, the enterotypes were nearly half-half. You see that it was 53% of PM enterotype, and the other one was RT enterotype. So from this base population, we derived 4 generations of selection. Jordi. Thank you. So you see that when you have nearly 1 generation per year, so the duration of the whole experiment was more than 5 years. So at each generation, we repeated the same protocol, collect feces on piglet at 60 days of age, sequence bacterial DNA, computed the relative abundances of each genera, and select the most interesting animals. And we go on like that for several generations. So the first result I want to show you is the evolution of the enterotype proportion. It's in each line at each generation. And you can see that really in both lines, the proportion of enterotypes diverge between the two lines. So in the HPM, the PM enterotype was nearly fixed at the fourth generation of selection. The percentage is nearly 100%. And in the other line, it was a little bit less efficient, but we have reason for this result, but you see that for the RT enterotype, we nearly reached 80%. So really, for a way to select for enterotypes, it was really efficient. We did great, in fact, for this, with a very parsimonious information, because relative genera of only 4 genera is quite, it's very limited information on microbiota, because in these animals, we identified more than 300 different genera, not all known, but the information is much more rich than what we have selected for. So, and we have estimated the heritability value, because it's the real parameter to see if selection can be efficient. And we find that several genera were heritable and with significant heritability, more than 0.1. And so that's the blue dots. And you see that the genera we have selected are at the higher range of heritability value, the most heritable being the T genus, the Treponema genus, sorry. The red dots represent the litter effect. So it's a statistical concept here when we estimated the genetic parameters, but it shows that the maternal effect is quite limited in our experiment. There were very few genera with what we consider a significant litter effect. So the only one was Streptococcus in our case. So the selection was not really driven by selection on maternal characteristic of the microbiota, but really the way we did it, and the response of selection is mainly due to the fact that we had a very high intensity of selection on the boars. So we also estimated some genetic correlation with a trait of interest. I just show you what we call post-weaning average daily gain. So in pig production, you have 3 different phases. The first one is milking period. So in our case, it's 4 weeks. You have a transition period. So and the main transition was the feeding transition from milking to solid food, and it's very important for the success of the beginning of the growth of the animal. And the last period is what we call the growing period, and it's between 10 weeks of age until slaughtering. So this period of post-weaning is really the transitional period for the piglets. And in our case, we see that there were some favorable correlation between Prevotella and Mitchokella relative abundances and average daily gain in this post-weaning period. So It was an expected result because we have seen before in previous studies that there were a link between the PM enterotype and this post-weaning growth period. So then we just to tell that our results are not completely different from other results in different studies, but in our case, we saw that measuring microbiota at this during post-weaning is quite better to estimate higher heritability values, because if for previous periods more in the milking part, the heritability are generally very low because it's really under the influence of the south, and in a later period the microbiota can be more diverse and under influences of environmental factors, which takes more importance. And in this case, the heritability is a little bit lower. Then a second result. So we show you the evolution of the enterotype percentage in each line, but we can also look at all the relative abundances of the genera. And here is the differences between the two lines over the fourth generation. So the gray, the light gray is the difference between the two lines in the first generation, and the black one is at the last generation of selection. So you see that, except for Mitsukela, the differences between the two lines was really increased by selection. So on the right part of the slide, you have what we observe in real life. So the black, the gray scale is for just the difference between the two lines. So it's you have the correction for all the environmental effects because the two lines were in the same environment at the same time. In the right, it's what we observe. It's the raw mean of each line at each generation of selection, and you see that the main response were in the RT line, HRT line, and not in the HPM line. But it's under environmental effects, so it was quite an interesting result in that case. So we also look at a bunch of genera. So there are 74 variables here. So it looks nice because the way we arrange the variables. But you see that on the left part you have all the genera that were relatively increased in the HPM line, and on the left part, and on the right part you have the genera enriched in the HRT line. It's not completely equilibrated between the right and the left, and it's linked to the fact that in the HRT line, the microbiota was enriched by selection, and we also observe the differences between diversity indexes between the two lines in the fact that one was enriched by selection, on contrary of the other line. And we look also at some production traits. The only one who was really affected by selection was average daily gain in post-weaning period. We also look at growth rate in the growing period, and in fact it's not affected by selection. So the animals that were heavier at a young age, when you look at the end of the period, of the growing period, there is no difference between the two lines, and it's impossible to see when you look at all the period if there is a better microbiota for the whole life of the pig. And you have Also, we look at the average daily gain in the milking period, and it's really also it's not affected by the selection. So the microbiota of the sow, which must have been selected also, is not influencing the growth of the piglet in milking period. And here you see the differences between the two diversity index in favor of the RT line. And we looked also at the function of the enterotypes to see if there are possible explanations for this divergence in post-weaning growth. So it's the number of animals is limited, but we can see that there are functions that were differently enriched in the PM enterotype or in the RT enterotype that can be related to this post-weaning growth period. And we also look at the species level, because with 16S we look only at the general level. In this case, we can dip a little bit on the species, and they are all related to Prevotella and Mitsuochela. And so, is it possible to select? Yes, it is. And if you want to know more, we have a very nice publication in microbiome. The question is another question we had is if we select on fecal microbiota, did we do something on internal microbiota? Because what is interesting, in fact, is not really fecal, but the gastrointestinal tract microbiota. So we slaughtered some animals at the third generation of selection. So these animals were 60 days old. And we observed that comparing animals from the two lines, we had a response on the microbiota from the colon with large differences and in the same way as the fecal microbiota, a little less at the ileal level, and nothing at the pulmonary level, which means that the genetic determinism of microbiota for really different organs or function is really not the same. So it was quite interesting, because it was one of our questions if the general host genetic, if we change a microbiota, our other microbiota are influenced by this kind of selection. And in fact, if it's a completely different organ, it's not. It doesn't seem to be influenced by selection. So we were quite happy that fecal selection can influence the digestive microbiota, because it's really the internal microbiota that has a function on the digestive tract, not the fecal one. Fecal one is just a proxy, but in our case, it's really an excellent proxy for this kind of selection. And two results, I will not be too long. So we look at some blood parameters in general, different blood parameters. So we look at the blood formula. We didn't see really differences between the lines, and also some blood immune response traits. And in that case, the most interesting result, there are several differences, but the really interesting one is on the T lymphocytes, because it's really related to the response, so the potential of the response of the animals. And in this case, you can see that the RT1 might be most responsive and more to infection, and could be favored in infectious context. Sorry. And the last, for something completely different, is we also observed metabolic metabolomics, but from the blood and the saliva. And here is the result on the saliva. And we observed at this stage no difference between saliva metabolites. But we have many things to look at in this on these two lines, but in fact we can see that we answer at least the question of the host influence on microbiota ecosystem, and we see that it's really possible to use genetic as a lever to influence ecosystem or microbiota composition. We can find easy proxy because fecal is really great for that, and it was quite challenging as an experiment, but it was really really nice results. So we did this work with a lot of people. So we have to thank really a lot of people, those who took care of the animals, who collected the samples, who did all the sequences and all the bioinformatics and data analysis. And Jordi will go on.
Yes. Okay. So this was the story about the pigs, but as we said at the beginning, there's also a short story that we want to share with you today on the cows. So what's happening in the cows? So it's a lot of studies are focusing on the rumen, but here we with a collaboration with a French company which is named Fusion and GD Biotech. So we had an industrial PhD, which we have here that's just defended PhD a few months ago. It's Louis Brulin. And in this case, we had access to the data they had generated in this company on 1,860 cows belonging to 140 commercial herds. And we had 16S sequencing and host genotypes on them. And as I was saying, we were focusing not on the rumen, but focusing first in the fecal microbiota composition. And we had observed here, for instance, that bifidobacterium abundance in the fecal microbiota was associated with milk traits in dairy cattle. And we also perform heritability estimates, and we observed that just in the pigs, just like in the pigs, that the fecal microbiota of Holstein cows has significant heritabilities, and in addition, it's also genetically correlated to dairy performances. So we could imagine that just like for the pigs, we could also try to use microbiota composition in the fecal samples of the cows to select or to direct selection. And not for today, but we also discuss results with gene diffusion on data we had on 16S in bovine vaginal microbiota. And in this case, we analyzed the results here, and we obtained associations between host fertility, longevity, health, and production by looking also at the vaginal microbiota. So each ecosystem is a story, and interestingly, every time we look, we tend to find interesting associations and in here. So a bit more far away on the Louis PhD with results that he just submitted to publication and expecting to get some answers. So we also perform genetic analysis for genome-wide association studies on the fecal microbiome. And here in the graph, you see the main results on the genetic associations and significant links with different taxa, and including Akkermansia, which is an interesting one, which is usually associated to better health in humans and candidate probiotics for humans in here. So this is the peak that was found on the chromosome 11 in the cow, and very interestingly, it was found on the top of the gene ABO, so the block group of for humans. And I think it's very interesting because it falls exactly in the same gene that was found associated with pig fecal microbiota in results published a couple of years ago in Nature. And in that case, they found an association with this family in here. but in our case, we found Akkermansia. So different species, but same genes having impact in composition of gut microbiota. And also there are results in humans that show that blood groups can impact gut microbiota. So maybe we can act on these genes to have interesting associations, in this case with Akkermansia, which is linked to health. So for a conclusion in the perspective, so we answered different questions in here. Is it possible to use host genetic selection to influence gut microbiota composition? Yes. And with this, we are also impacting other ecosystems and host traits. And then the next more difficult question is if it's going to be useful, because we can do it, but it's going to be of use for the production and livestock industry. So for this, so we have to take this holobind concept in animal genetics perspective and try to include the microbiota composition in this formula that phenotype equals to genotype plus environment plus M on the microbiota composition. And we could imagine ways of doing this efficiently and to contribute to improve the efficiency of genetic selection in livestock. So, but for this, there are quite a lot of challenges that are upcoming. Some of them that may be shared even with the plant genetics community. And the first one is to define the cost effectiveness of this microbiome-informed breeding and selection programs. So is it worth doing it if it's cost effective for doing it? And we need also to update the statistical and modeling approaches that are necessary to integrate this type of data. So there are a lot of efforts in the moment towards in this direction by integrating not only microbiota but many type of omics in the genomic selection, for instance. And then we need to go back to biology, because we have seen there were different results in function of the ecosystem, function of the species. So we need to define probably for each character, for each trait of interest, which microbiome to sample, when to sample this microbiome, and also learn what are the microbiomes are actually doing. So with meta-transcriptomes or meta-proteomics analysis, and we need to define if this is useful for all host traits, or if it's maybe worth going for traits that are most difficult to measure, like for instance immunity, health related traits, as we are trying to do in the case of the big story that we presented today. So, and finally, we will need to do a lot of more of experiments. So we need another letter in the equation, the euro from the E, to try to fund all these questions in the future. So that was all for the story I want to talk to you today, and just to let you know that in the summer in 20 to 25 July, we have the ISAC conference 2025 happening in Daejeon in Republic of Korea, and if you are interested to come, so we will have a microbiome session dedicated to livestock metagenome analysis, and we will have an update of the results that we're presenting here, and also many other interesting talks that are scheduled for this. So with this, I give the floor to Claire for the questions and answers to be answered in the presentation.
So thank you very much for the very interesting results. And we have few questions in the Q&A session. So first, maybe Catherine, it's for you, but of course I know Jordi, you could answer also. So the first question is, maybe you have to better explain the genera that were used for the selection experiment. I read the question, why were different genera evaluated for the different pig lines? And I assume that this would have introduced heterogeneity in the experiment. One expects that the two lines should be as homogeneous as possible. Your explanation for this will be appreciated. So maybe you have to explain what was measured and that for assessing the microbiome of the two lines.
Yes, we could have made a selection with two lines really divergent for one genera or two. So it would have been Prevotella in this, because it's the most abundant genus in in the feces of the pigs. The answer to that is because it has never done before. It has never been done before. So we were quite confident it would work, but it was quite a challenge at the beginning. So to increase the chance to see divergence or differences between the two lines, we think the selection more like enterotype selection than really one genus selection. That's why we have chosen these 4 genera. The real selection was on 2 genera because it's quite difficult when you know not enough genetic parameters to select on 4 genera in a really strict way. So it was a kind of balance, but the main genera selected were Prevotella for the HPM line and Treponema for the HRT line. But this was really driven by this idea of enterotypes. That was the main concept driver of this selection, and that's why we selected on different genera in the 2 lines.
So thank you. So there is another question, and maybe Catherine, you can answer very quickly, because the question is, were pigs from the different selected lines co-housed, or could there have been a reduction in exposure to microbiomes of one enterotype at the selection experiment as the selection experiment progressed?
They were co-housed in the same building, but they were not co-housed in the same pens because pigs are raised in pens of so in our case 20 animals in post-weaning period and 10 animals in growing period. So the 20 animals in the pen were from the same line. Why that? Because we wanted the selection to work. So we didn't take too much risk by mixing everything. So we've chosen to have the animals, the 2 lines in the same building, but not in the same pens. But we have small protocols where we mix the lines. We don't have the result yet, but we did that.
Maybe Jordi, now it's for you. When you select for different microbiota profile based using the host genome, if you change the environment or the food, how much of the selected microbiome profile will be kept? So it's a connection with the environment.
That's also a question that we're working on, but the Larzul present we're really focusing on the divergent selection and to create the 2 lines. And now we are exploring this by asking different questions on these animals. And for the question you're asking here, so we have been doing it's to inseminate sows in different environments with the boards that we have kept the semen. And so we are going to see if the differences are still there with animals raised in organic production system in a different farm, and also in in-rye farm that is located in the Caribbean. So we'll see if in tropical conditions both lines are different for the phenotypes and also for the microbiome composition. So it's it's a very good question, and we're trying to answer to this with new results.
And maybe again for you, Jordi, for the GWAS on fecal microbiome in cattle, what was the sample size used? And other comment, is the publication available?
Yes, so the publication, we hope it's going to be available very fast because it's in the 2nd round of evaluation in a journal. So hopefully very fast you will have these results. And then for the sample size, it was the 1,800 individuals. So it's not a big number for a usual traits analysis now in cows, but still I think it's one of the biggest GWAS analysis for microbiota composition in cows. That's for sure. So that's why we were not observing huge peaks, but we are quite confident that the peaks we observe, they are the real associations. And the fact that it falls in a gene that is associated in all the species also with microbiota, it makes us confident on the result we obtain.
And then maybe there is one question, and both of you may answer, and you choose who starts. If certain microbiota do not positively influence the health and productivity of the host, which strategy is more appropriate to implement? Targeted drug intervention or selective breeding? So how to modify your microbiota, maybe that is not perfect for either health or productivity.
Jordi.
I go. Okay. So I think it depends on if you are looking at a short-term or long-term scale. If you want to influence animals that you have already in a farm setting, you cannot use genetics for this. So you can use the diets, you can use probiotics, you can use management strategies to try to drive microbiomes towards a benefit, a good composition. Even you need to still to define which is a good composition, which is a question on itself. But you have all the nutritional strategies to go for this. But then if it's in the long term, you identify that it's a positive association each time that with a given microbe or given composition, then it makes sense to evaluate if it's interesting to use genetics for this. So you need to have a good heritability for this microbe, and if you have this, you could implement selection strategy. But as I was saying, it relies a lot on the cost effectiveness on the approach also. Because if you are selecting for one trait, you are probably less effective for another trait. So you need to put this in balance. Maybe Catherine, you can comment on this.
Yes, first in our experiment, we didn't measure a lot of traits because it was basically a concept proof of selection. It was not to explore all the potential of selection of microbiota, of selection of the host on microbiota, because first we have to prove that it works. So we put all our effort in the selection and not at all the potential response on the health trait. But we have indication that it has influenced traits not at all related. For example, on behavior, the RT line is much more active, nearly aggressive than the other lines. So this result was not expected. So we are trying to analyze it correctly by video recording, but from the animal caretaker, they really observed that at young age and later on, the behavior of the pig was modified, and they can observe differences between the two lines. So on health, we don't have a really challenging environment. It's the rules of biosecurity are quite high because of we have to preserve the selected lines. So we try not to challenge too much the animals. They have to reproduce at some point. But we are evaluating the lines in completely different environments. So we have offspring of the lines in an organic system, which is completely different with a high fiber feed and a longer milking period and so on. So we are looking at that, and we will also have evaluation in a tropical environment to see if for heat resistance or things like that. If also we have differences, and even if the composition of the microbiota is kept in different environments, and if we still see the differences between the two lines.
So thank you. So there is one question. Have you studied how adding microbial-based products to feed or on grazing lands impact the gut microbiota? And that is important because you have the influence of the farm practices and the impact also of the plant environment, natural or provided by the feed.
No, we didn't.
Not in these animals, but then there are extensive literature on colleagues working in nutrition that look at the effects of diets and different sources of diets, so different plants or different composition of fibers, and they found very consistently that it impacts microbiota composition. So that's for sure. There is a very effective way of modifying microbiota composition is through diet.
But for the experiment you reported on cattle with the genetics, the host genetics on cattle, how were the animals fed, for instance?
In this case, there were 140 different farms. So each farm they had their own feeding, and this was included in the model as a farm effect. So we are having different diet, different condition, different management, and they are all absorbed in the farm effect. So the design was not really think as a way to try to challenge the question of the diet. It was really more on the we saw a very different set of environments, which is the impact of host genetics, and we saw that there are significant effects in there.
Maybe I'm not sure to understand, Kelly, your last question on products on plants subsequently used for feed. Maybe if you can precise the question, I can ask it to Jordi and Catherine. Actually, I could follow up on the previous questions. How do you foresee you see in this perspective of interconnectedness between plants, animals, soil? How do you foresee work in animal microbiomes to feed this new field of research?
Well, if I had to say something, I think if it's on the circulation of microbes from the fields to the feed for animals to fecal samples of animals that then can be used to fertilize the fields again. So there, it's probably synergies to identify in there and how to make or at least make us aware of the relevance of the circulation and maybe identify sources of improvement in here. So you can explore this side of the circulation, and then I think it's a lot of work to be done. One in the question that was asked also before, in the variability of the plant sources and the plant within plant feed, and select animal feed plants that can be used for to improve feeding and efficiency of the whole system. So there's also genetic selection on the animals and genetic selection for plants directly for animal feed. So this breeding in this case could be useful, and it could be made more efficient also taking into account the microbiomes of animals, microbiomes of the soil, and how they impact the host plant and host animal.
And actually, are you aware of any experiments that would assess the quality of the manure according to the diversity of the animal microbiomes?
Not aware of that. I'm aware of a lot of research is done, but it's on the for the production of methane from animal manure, and probably there is also a question in here on the quality of this manure or how to implement better increase the efficiency of methane production in the methanizers, which is the opposite question that for ruminants we want to reduce the production of methane, but in the bioreactors we want to increase. So I think there is a dialogue between these communities, essentially the ones working in ruminants and the ones working in methane production in bioreactors. But for the quality of the manure for the field, I think this would be a very interesting question. So if somebody is interested in this type of approach, we will be happy to listen and to learn from them.
So we have a clarification for one question, because the question was you see how to add products on grazing systems. So I read what is written. I was really trying to get at whether adding biological products to grazing land could impact microbiota. And it would not be an intentional diet supplement, but rather a farming practice to improve soil or plant quality. So just to know if it is something that is in the scope, looking at work on rumen microbiota studies.
Yeah, I know, Catherine, if you have an opinion on this.
No,
I
but
think it's a question. It's a question
vision
for
of
grazing, for ruminants. I would say it's a major question in this case for meat production in ruminants, probably.
So that's still an open question. Maybe Catherine, do you want to comment?
It's for